We've talked about meiosis, and we understand that meiosis occurs in order to facilitate sexual reproduction. One thing that we want to now mention is that the biggest advantage of sexual reproduction, as opposed to asexual reproduction, is that it introduces genetic variation into the offspring. And this is a major theme in biology, and something we'll talk about over and over again throughout the course of our class. And so now we want to ask the question, how does sexual reproduction introduce variation, genetic variation that is, into the offspring? And to do so, we need to make sure we understand the concept of homologous chromosomes. I hope that we do from earlier in our class, or rather from earlier in our lecture in this chapter. But I want to make sure that we do, and so I want to look at our diagram of homologous chromosomes that we have on the screen. These homologous chromosomes, like all homologous chromosomes, contain the same genes but different versions of the same gene, and we refer to those different versions of the same gene as alleles, a term that appears on this slide. For instance, here's a flower, and in this particular plant we have two chromosomes that are part of that one homologous pair. And at this particular part of the chromosome, this location, something we refer to as a loci, L-O-C-I, on the chromosome, then we have a gene for flowers. But on this chromosome, we have a gene for purple flowers, and on this chromosome, we have a gene for white flowers. And so we refer to those as alleles because they are the same gene, but they're different forms of the same gene. Okay, this is essential to understand to talk about what we're going to talk about next. Crossing over is that process that occurs back in prophase of meiosis 1, and in crossing over, the homologous pairs of chromosomes come together, they become zippered together via this protein lattice, and they swap segments. They literally trade segments. And so we can see that occurring right here if I point to this portion of our diagram from our text. So these two arms of these sister chromatids come together, and part of the blue chromosome ends up on the red one, part of the red one ends up here on the blue one. And so of course these chromosomes represent the red one, say, came from the mother, and the blue one came from the father, and so they literally swap segments. And so now on the chromosome from the father there's part of the mother's chromosome, and on the mother's chromosome there's part of the father's. And remember, these are going to split at meiosis, and each of them are going to go to an egg if it's a female or a sperm if it's a male. And so interestingly, crossing over occurs, and crossing over is a mechanism that scrambles the genes before they're passed on to offspring. So it's interesting to think about, perhaps disturbing, but if you think about it in these terms, then crossing over occurred in your parents, and your grandparents' genes were scrambled in your parents before they were passed on to you. And so interestingly, this process of crossing over introduces a great deal of genetic variation into the offspring. Crossing over always occurs, not just sometimes, but always occurs. And as you can see very clearly, it works simply because of the nature of homologous chromosomes. The same genes are located at the same point on the chromosomes, and so when you swap segments, then you end up with the same DNA. Now you may very well have a different allele, but nonetheless it's the same gene because the genes are located at equivalent points on the chromosomes. There's one other way that genetic variation is introduced into offspring via sexual reproduction, and that's something called random alignment or random assortment sometimes. And so what this means is as follows. If you think back to meiosis 1 and metaphase of meiosis 1, then you remember that the chromosomes line up along the metaphase plate. And so when the chromosomes line up, there is a maternal chromosome and a paternal chromosome, and they have to end up on either the left-hand or right-hand side of that metaphase plate. And so what random assortment or random alignment says is that there's a 50-50 chance that one chromosome will end up on one side of that plate or on the other side of the plate. There's no rhyme or reason to how that occurs. And additionally, this is the most important thing, the way that the first chromosome lines up has nothing to do with the way that the second chromosome lines up. So let's look at a diagram that perhaps will help us with this idea. Here we see two chromosomes. The red ones came from the mother, the blue ones came from the father. This is metaphase of meiosis 1. And so in this case, in this hypothetical example, then the red chromosomes have lined up on the top, the blue ones on the bottom, and so the red ones are going to eventually end up into two in cells together, and the blue ones will end up in cells together. But what we're saying here is that the way that the first pair lines up, in this case the red one on top and blue one on the bottom, maternal on top and paternal on the bottom, has nothing to do with the way that the second pair lines up. And so the first alignment that we have here is possible, but the second alignment is also possible. And so interestingly, there is this astounding number of possible genetic combinations that comes as a result of this process. If you think about human chromosomes, we have 23 pairs, and so the first pair lines up and all the other pairs line up independently of that. And so you are the result of this process. You are this interesting mix of chromosomes that came from your parents. Of course, those chromosomes came from your grandparents. The only way they got into your parents was from your grandparents. But when they were passed on to you from your parents, then those chromosomes lined up, and you didn't get all the chromosomes, say, from your grandmother or from your grandfather, but you got a mix of the two as a result of this process of random assortment or random alignment. So interestingly, these two processes, random assortment and random alignment, introduce a great deal of genetic variation into the offspring. The genes that we see on the screen here, in case you're wondering, they're not especially important to understanding this idea of random alignment, but your book uses them as an example of two genes that are very serious. HER2 is a gene that's associated with ovarian cancer, and BRCA2 is associated with breast cancer. And so BRCA2 positive means that you have this mutation that will increase the likelihood you'll get breast cancer, and HRE2 same way, and then the minus indicates that you don't have that. It's also interesting to note that the gene that Angelina Jolie carried was BRCA1, and so that's what led her to get that radical procedure done that I mentioned earlier in our discussion. All right, and so that's how genetic variation. Those are the two primary ways that genetic variation is introduced into offspring. First, we have crossing over, and then we have this idea of random assortment.